All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Today we have a topic from cancer or oncology, and this is an important topic. There is a new study that just came out. Although the study is old, it is from 2019, but it just got accepted and published. So let's look at this study. The basic summary of the study is that they have found, the researchers had found a new cell type that is neither acquired type nor innate type. It works like acquired. It also works like innate. It's a beautiful cell. If that cell was big enough, I would have hugged it. So that is the cell which they feel or they have discovered that it works to take care of the cancer cells, especially the solid tumors in our body. And they found out how to activate it. That is the big important message today. Interleukin 15 is used to activate it. Number one. Number two, they found out that when other immune cells, other T cells become exhausted, this cell is not exhaustible. It cannot be exhausted, and I'll show you why it cannot be exhausted. So this is a perpetually moving, of course, perpetual motion machine is not possible, but this is a non-exhausting, continuing to work cell, and they have figured out how to activate it against the tumor cells. And so there are high hopes with this, and we are saying that there is a gap when we do immunotherapy of cancer patients. There is a gap between successful therapy and the immune system's activity. And it turns out that this cell might be that bridge that will bring immune system even further towards the successful immunotherapy. Let's start this. I would keep singing the praises if we didn't start looking into it. So this is a gift for humanity. This research is done by from Memorial Sloan Kettering Cancer Center, that is in New York, and Ming O Lee and his team are the ones who worked on it. Let me just very quickly show you the paper. This is a paper. I cannot go over the whole paper, although I did work on it. The reason is it is a subscription-based paper, so this is a document that is to be purchased, so I cannot show it to you. So my apologies, but I'll present what they're talking about in here. So that is one. Then here is the quick note. This is drbeen.com. In the link, there is in the description, there is a link to buy Dr. Bean at a very affordable price. This is CDC talking about the top reasons for death in older age, and cardiovascular problems and cancers are the most important thing. So if you see here, leading causes of death among persons aged 65 and over, heart disease, cancer, and now COVID-19 as well. This was a previous document from CDC as well. And here too, they talk about the top causes of death as we get older in the US are cardiovascular and cancer. And in the US and in many of the developed world, we have taken care of majority of the infectious diseases. So what are left are the diseases with cardiovascular system, respiratory system, cancers, and so on. This is 2016 study by the same researchers when they found this cell. So cancer immunosurveillance by tissue resident innate lymphoid cells and innate-like T cells. So this innate-like T cells is what we're going to talk about today. So when they found this cell out in 2016, what they did not know was, where does this cell come from? What is the origin of the cell? And secondly, what is the function? How do we make it work for us? That was not clear. So it just became that a new cell is discovered. Then here, this study was actually submitted in 2019. And now it got published April 2022. And this is a program of self-reactive innate-like T-cell mediated cancer immunity. So they are actually showing you the program. They actually took modern apparatuses, for example, CRISPR technology and the single cell analysis technologies, and looked into the cell and then found out how the genes are expressing and what the cell is doing. And so they are able to share the program of this cell. How does it work? So that is a beautiful thing. And then all the remaining links are in the description as well. They are interesting and related to this. So with this, let's start our discussion. So here is the summary. On this page, I'm going to try to provide you the whole summary, and then we'll go in the details later on. Maybe this and the couple of next slides. So first, let's look at this diagram. Here, these cells that are under the feet of this cell, these cells are, and you are going to have some fun with the diagrams today. I took a lot of time today to draw them for you. So these are tumor cells. On the tumor cell, the cancer cells, is this new kind of a cell. This new kind of a cell is neither acquired in its properties, not innate in its properties. Instead, it is actually a combination of both. What is the properties of this cell? Number one, this cell has T cell receptors on it. So because of the T cell receptors, you can call it a T cell. And you can call it similar to cytotoxic T cell or CD8 T cell. But I would explain later on that it doesn't behave like CD8 T cell. It actually behaves like innate cells, but it has receptors for T cell. This is why it is called a T cell. And it also has this receptor on it for immunoglobulin. So this is FC, remember fraction constant, FCER1G. So this is a receptor which is unique to this cell, and that receptor is for immunoglobulin E. In addition to this, it has granzymes on it as well. And we know that natural killer cells, for example, and cytotoxic T cells, they use perforins and granzymes to kill the cell or pathogen that they are fighting with. And they called it ILTCK. Very interesting. Innate-like T cell. So innate-like means innate arm cell, which would not need any other cell to help it. It would just work by itself. Innate-like, and I'll explain that a little more. But at this time, from a bigger level, innate-like means something that belongs to innate cell. T cell, something that belongs to acquired cell. Killer, something that belongs to innate arm. And the acquired arm. Cytotoxic T cells are killers, and they are part of acquired. And natural killer cells are actually part of the acquired that have gone into the innate arm, just like this cell as well. So you could actually call it a natural killer cell-like cell, but it still is not like them. And its activation is with interleukin 15. It's a fascinating cell. Now let's look at the summary. It has alpha beta T cell receptors. So it is T cell receptor positive cell. It has FCER1G expression, which is the immunoglobulin E receptor on it, which is unique to this and the other natural killer cells. It behaves like innate cells. What does that mean? Innate cells, macrophages, dendritic cells, neutrophils, natural killer cells, they do not need another cell to activate them. They can by themselves, all by themselves, sense a foreign antigen or a problem in the tissue, for example, cancer cell, and they can by themselves attack it. If you look at the acquired immune system's function, over there, some cell has to come and activate them, has to present an antigen to them, then release some cytokines to activate them. So I call it hugs and kisses. They need lots of hugs and kisses to work and become activated. But innate arm does not need it. These cells by themselves are enough. So this cell by itself is sufficient, and it can become activated without the need of an antigen presenting cell to present it an antigen. It can sense the antigen and just attack it. It is highly cytotoxic as well. That is, it is a killer cell. It is like cytotoxic T cell, CD8 positive, or natural killer cell. They both are killer cells. This is also a killer cell. And it is found in malignant tissues. It's found active in malignant tissue. Otherwise, it is present everywhere in our body. But it is not normally going on killing tissues. It is just sitting there. But in malignancy, when the tumor is formed, then the tumor cells release interleukin 15, and that interleukin 15 activated, and it starts killing the tumor cells. So, of course, as you can know that that many cancers are not controlled by our body. So, of course, this means this cell is not always successful. But the researchers are saying we can actually make it successful by genetically engineering it or by providing interleukin 15 to the patient. And interleukin 15 will activate it in the tumor area and it would attack those cells. This was discovered in 2016, as I said before. Now, it is very important part here. Reactive to unmutated self-antigen. This is another unique property. And I would go in detail a little later. But here in the summary, a cell that attacks our body, an immune cell that attacks our body, is not a good cell. We usually, as soon as that cell is born, we usually identify it in thymus and we kill it. We don't let such cells go in the body, although such cells do escape and cause autoimmune diseases. Here we are going to look at a cell that actually attacks our body tissue, still escapes the thymus, and becomes very, very useful in the future if we have cancer developing anywhere. This is not the situation with the other tumor killing cells. For example, CD8 cells or natural killer cells, they usually do not attack our cells' unmutated antigens, meaning normal cells. Then this cell can continue to replenish. That is another thing. It just keeps, new cells keep coming in, they keep replenishing. And then this cell is non-exhaustible. It has specific, imagine if you have a car and that car has brakes on it. In the immunology world, those brakes will be called PED, programmed death molecule 1, and CTLA4. And PD and CTLA4 are disabled on this cell. So imagine this is a cell without brakes. You cannot stop it when it is active. It cannot be exhausted. And that is a beautiful property. And we'll talk more about it. So I want to go back here and kind of complete our summary. Actually, there's one more point here as well. These cells, these innate like T cell receptor or T cell positive killer cells, they have another very interesting property to them. We are still at the summary. They have another very interesting property. If there is a tissue, they actually go deep in it. Other immune system cells that are natural killer type cells or that are killer type cells, usually they circulate in the body, they go to the lymph nodes, they go to the blood, then they go in the tissues as well, but they usually do not go deep in the tissue. This cell can penetrate deep in the tissue and go and fight with the cells, the cancer cells. So I'm going to quickly do a summary here. So let's say somebody has cancer and you are going to provide them some information to say, please check this out. Number one, the patent for this technology now is with the Sloan Kettering Cancer Center, New York. They have the patent on it. So contacting them will be interesting. This is not a sponsored video. Secondly, interleukin 15 is the important molecule that activates these cells. So that is also another important thing to keep in mind. Number three, these cells are actually present all the time in our body. They are here. and they become activated when the tumor develops. And who knows how many times they become activated and take care of the tumor and then move on. But if the tumor becomes bigger, it is possible, according to the authors, to potentially give interleukin 15 or engineer more cells of this type, and that would help suppress the tumor. In their own experiment, which they did in mice, when they gave interleukin 15 to mice with cancers, the cancer growth was suppressed. When they did not give interleukin 15, the cancer continued to grow. And the reason was that these cells became active with the interleukin 15. Then they said maybe this is a study in the mice, so maybe this is a property of the cells in the mice. So they then took human cancer cells, for example, colon cancer cells, and they identified these cells in the colon cancers as well. That means these cells are present in us as well, and they become active as well with interleukin 15 in the cancer cells. So that is the summary. I'm now going to go into the detail, a little more detail. The whole paper is so technical and it's beautifully written, and I love immunology, so I just enjoyed it. But presenting that over here would take probably a couple of hours. So I'm going to go in another 30 minutes or 20 minutes of the fascinating characteristics of this cell. So many of you would probably already know this cell. If you did not, I am proud that we are learning about this cell together. This is a cutting edge moment for this technology. So here, non-exhaustible cell, PD1 and CTLA4, and I would explain that a little later, goes deep in the tissue as well. It actually lives deeper. So here, the first explanation, why is it not exhaustible? When other T cells become exhausted, why does this not become exhausted? So let's start. There are two mechanisms. Imagine that this is a cancer cell. Here, this guy is a cancer cell. Cancer cells are very smart. This is why they can outcompete with the other cells and keep growing and cause cancer and kill us. They have lots of tricks under their sleeve. One of the things is that they do is that they develop this protein on their surface called PD ligand 1. And here, imagine this is a T cell. What happens is this T cell, let's say this is cytotoxic T cell, this T cell comes and connects with the cancer cell, because cancer cell is actually a cell, right? So it is presenting the antigens, the transformed antigens. The cancerous cell has cancerous proteins inside, or kind of abnormal proteins. And so those pieces of proteins are now being displayed on the cancer cell. Antigen is being presented. The T cell comes in, connects with the T cell receptor to this antigen. And this antigen is being presented on, so for medical students, nursing students, doctors, NPs, PS, DNPs, MHC1, histocompatibility complex 1, that is for all the nucleated cells. So MHC1 is demonstrating or displaying the antigen, and the cytotoxic T cell is now connecting to this antigen through T cell receptor. We know that there is now going to be co-stimulations that are needed. However, the cancer cell has PDL1. So what happens is this T cell has a molecule on its surface called program death 1. If you stimulate this molecule, if you shake its hand and say, leave us alone, then it would leave you alone. So it has a special hand for that, to take this little bribe from the other cell. So PD1 is a molecule that is present in the surface. If that molecule is connected with PD ligand 1, then this connection will cause this cell to become exhausted or become non-responsive, even when a response is needed. Remember, the definition of exhaustion is different from definition of inactivity or activity. Exhaustion is that we need this to be active, but it has decided, because of the environment or because of these signals here, it has decided not to respond. We wanted it to respond. We needed it to respond. Its response was needed, and it is not responding. So that is the exhaustion. So now here are a bunch of exhausted T cells lying around because they're not attacking the tumor cells, because tumor cells have pressed their brakes. Another similar mechanism happens with the antigen presenting cells. For example, now these are professional antigen presenting cells. These are macrophages or dendritic cells. So what is their function? Macrophage or dendritic cell will come in and find a cancerous cell. It would eat it up, the cancerous cell, and break it down and then present those antigens on its surface, or not surface, with the MHC2, major histocompatibility complex 2. Now here is a T cell. T cell would connect with this through the T cell receptor and identify that there is an antigen. That would mean that this T cell needs to become active. However, macrophages can also decide that, hey man, don't become active. It's okay. This is our own tissue. Don't destroy this. So macrophages have molecules which are called CTLA4 or CD28 and they can engage these molecules with the T cell, with the surface molecules on the T cell, which are called B71 and B72. Generally, once again, macrophages have also a mechanism to press the brakes on the T cell's activity and not let them be activated. Why would a macrophage do that? A macrophage would do that when it thinks that the antigen it just picked up is ourself. So maybe let's say a macrophage is killing another macrophage that has died. So it's not killing it, it's clearing it. So now, of course, this macrophage should not activate the immune system because that is self and it is just the trash need to be removed. So in such case, macrophage will not want a T cell to become active. Otherwise, unnecessary inflammation would start. So here, this macrophage is inactivating even after the macrophage has actually picked up a cancerous cell. Why is that? Why will a macrophage or a dendritic cell stop a T cell from becoming active even after picking up a cancerous cell? The reason is cancerous cells are such enigmatic cells. They are on one end cancerous, that is, they're bad. On the other end, they are self. They are our tissue, our cells that have become bad. So our immune system has a real hard time to figuring this out, that on one end, that cell looks like our cell because it is our cell. So the surface antigen, the structure is all self. So we have to let it go. We have to be kind to it. On the other hand, that cell has become bad and it is now cancerous and it is going to grow. So we need to kill it as well. So our immune system sometimes cannot really figure out what to do in such situation and can incorrectly inactivate itself by thinking that, hey, this is a cell which is our own. Why should we attack it? So then, of course, this means there are therapies, there are chemotherapies or cancer therapies where we give blockers of these molecules. For example, if you give a blocker, these are called the inhibitors for the exhaustion. So what happens is, if you give PD1 blocker, for example, that would block this PD1 and not let it connect with PDL1. And so this cytotoxic cell will then not become exhausted. Similarly, we can block the other side, PDL1, ligand 1. And if you block that, again, this bond will not occur and exhaustion would not occur. In some patients it works and in some patients it fails. Same thing over here. One can give CTLA4 blockers, or one can give B71, B72 blockers and kind of block this bond from occurring. And sometimes it works and sometimes it does not. Now, how about if we have a killer cell? If we have a killer cell which actually does not have these breaks, which doesn't have PD1 and which doesn't have CTLA4, it would just by nature not blockable. So, continuing, this cell, as I said before, was discovered in 2016 by the same team. But at that time, they did not know what is the origin of this, what is the function of this, how do we activate it. So then now they went again after this cell with more sophisticated technologies. And for example, they had CRISPR. With the CRISPR, they could actually see what genes are being expressed. And they could kind of play with the genetic expressions and learn. And similarly, they had the technologies for single cell analysis as well. Here we have this cell, which is this cell we knew in 2016, but we didn't know what to do with it. And now they started understanding how does it work, what is the importance of interleukin 15. Then they wanted to understand, does it look like a CD8 positive cell or does it look like a natural killer cell? And they figured out that it is very different in its expression. It has similarities with both, but it is like none of them. So they said this is a new cell. It is partly like innate, it is partly like T cell. How is it innate? It does not need, this cell does not need this behavior that something has to be presented to it. It doesn't need that. It would just work with the cancer cell and sense that the cancer cell has abnormal proteins on its surface, and just presence of interleukin 15 is enough for it to become active. So that is an innate like behavior. On the other hand, it has T cell receptors on it. That is a T cell acquired arm like behavior. So they learned more about this. Then here is another important thing. This innate like IL T cell killer cell, they're not calling it natural killer, they're calling it IL T cell killer cell because it is kind of a hybrid. It recognizes mutated and non-mutated protein. This is a very important thing. You might actually say, well, so what? Okay, fine. Some technical thing Mubin said. No. Other cells, natural killer cell or cytotoxic T cell, they usually only identify abnormal proteins on cancer cells and then connect with them or then become active against them. This cell can actually identify a broader set of proteins, even if they are not abnormal by the cancer. So if this is a cancer cell, this cancer cell is going to have many kinds of proteins on its surface, some normal. Remember, this was a normal cell before, and some abnormal. This T cell, this killer innate like T cell receptor positive killer cell, can actually work with a broader range of proteins on the surface. That means it can become activated with more self tissues. So then you should have a question. If it can become activated against normal self antigens or proteins, then why is it not always attacking us? Right? We are saying here, unlike other cells, cytotoxic T cell or the NK cell, natural killer cells, which need abnormal proteins on the surface of a cancer cell, and they work with those abnormal proteins to recognize them and then attack the cell. Unlike them, this cell, this new cell, can actually work with abnormal proteins, but it can actually work with normal proteins as well. So then the question should arise that if it works with the normal protein, then why is it not always attacking us? Because our cells have normal proteins on their surface. So first question is, why did it escape thymus? Thymus's job, remember this is a T cell receptor cell, so it has to go into thymus to get trained. It's a T cell, T for thymus, right? How can it escape thymus? And in such large numbers. Every once in a while, thymus can make a mistake and we can end up with an autoimmune disease. But here we are talking about a cell that actually always escapes the thymus and goes and lives in the tissues. The question is, how does it escape the thymus? And the researchers found, so let's say this is a thymus. Thymus has a very interesting situation. The T cells enter the thymus. Imagine thymus is a school. Naive T cells enter the thymus. When they enter it, inside the thymus, there are cells and presenting cells like macrophages, dendritic, those kind of cells, or just simply presenting cells. They are standing there, as you can see here, and they are doing two things. Number one, they are trying to handshake with this T cell. They're trying to connect with this T cell with MHCs, major histocompatibility complex. If this T cell will not connect with them, if it will not handshake with them on MHC molecules, then it will be killed. Why? Because majority of the immune function in our body that would occur with the antigens is going to be with MHC 1 or 2 molecules. So if this T cell is not able to connect with that molecule, then it's a useless T cell. How would it work with the immune system? It cannot even handshake with them. Now, if it connects with major histocompatibility complex molecules or MHC 1 and 2, then the next test for this little afraid T cell, all T cells that are passing through here, is going to be that these cells, these presenting cells, will present an antigen that is self to this cell and say, what do you think about this? So remember these little, look at these black things. They're all holding some antigen. Everyone has a different antigen on their surface, and they are presenting it to this T cell. These are all antigens that are self antigens. Ideally, now this T cell should ignore it. If this T cell accidentally or deliberately connected with this antigen and became active, then this will be killed right now, right here. Because if this gets out, it's going to start attacking our body. So it's a very tough school for this T cell. If you don't do a handshake, you're dead. If you incorrectly attack the antigen that is ourself, you're dead. So how come this T cell, this new type, that actually attacks our tissue, how come that gets out of here? So researchers found that this cell actually cannot attack until interleukin 15 is present. So when it is passing through this school and it is being tested against the self-antigen, although it has the capability of recognizing the self-antigen and attacking, it is just not active because it doesn't have interleukin 15 present. So because it is not active, it is kind of keeping its head down and getting through this side, the thymus, and getting out of it. That's a beautiful mechanism. So it is positive selection and negative selections are the terms that we use for the teaching here in the thymus. And it actually gets properly selected and then comes out. Then it goes to the tissues. So that is the first milestone it has accomplished, and that is it passed through the thymus. Then it goes now into the tissue and start living there. And researchers said that unlike other, unlike other immune cells, which live in the lymph nodes, circulate in the circulation, live in some tissues as well, this one doesn't do all those things. It actually goes directly into the tissues and start living there. They said it homes into the tissue. So now imagine these guys are these new cells. They've gone and they're living in the tissues, and they are living in all of our tissues. When some cell or cells in our tissue, normally it is one cell that becomes mutated and becomes cancerous, then that one cell keeps dividing and proliferating And that is how the tumor develops. When the tumor starts developing, tumor releases interleukin 15. That activates these dormant neighbors who would not do anything to our tissue. But as soon as that interleukin 15 is present, they're going to start looking at the tissue and start attacking it, including, of course, the main culprits, the cancer cells. Now, of course, this would be the next question, and that is, when these cells are present in the normal healthy tissue and they have the capability of attacking the tissue, then why are they not attacking? And the answer is, normally in healthy individuals and healthy tissues, interleukin 15 is not presented, not created, not produced in high enough amounts to activate these cells. But when the cells become cancerous, they start producing lots of interleukin 15, and that is when this become active. I believe that is the end of the talk today. So what did we learn about? What is the practical outcome? Number one, this is a patented cell now and its procedure. So one has to connect with those hospitals, the Memorial Sloan Kettering Hospital in New York. Secondly, it is interleukin 15 that can activate this new kind of a cell, and that can help against solid tumors. And the researchers saw that if they give interleukin 15, why am I repeating this? Because it is important for us to know. Again, the second major cause of death in the older age is cancers. We all, at some point, are going to be nervous about cancers. So this is why I'm kind of saying it again and again. Interleukin 15 activates this cell, and this cell would then help with these solid tumors. The researchers confirmed this in mice, that they had the mice with the tumors, and when they added interleukin 15, the tumor was suppressed, the growth was suppressed. Those mice that had cancer and they were not given interleukin 15, their cancer continued to grow. So this is the discussion. Thank you very much for watching it. There are a few links in the description. If you like this kind of a work, and if you would like to watch more such lectures, there is a link in the description, which is, I think, not even 2 lunch amount. And that gives you access to drbeen.com premium content over there. There are 8 to 1,000 videos sitting there. And we keep adding new videos every week, and they are part of the same price. So you don't have to pay more. So I think that's a beautiful offer. And then in addition to that, there are links if you would like to support this work. You can become a patron. You can buy me a coffee, or you can use PayPal. Now there is a Substack link in there as well, and you can become a Substack member as well. Thank you very much. And I can now hang up, and I will see you on the Chit Chat channel in a few minutes. Bye-bye for now.